Excellent. Well, hello again, everybody. Welcome to another session of our virtual global spine conference. We are very happy tonight to welcome Shambhavi Bhagwat, who's my PhD student here at WashU, and you're going to hear a little bit more about her in a second. I'm going to just give some introductory slides here, and and then I'm going to turn it over to her. So I'm going to go for about 15 minutes with a little intro, and then turn it over to her. It's going to be a little bit different tonight. It's going to be more of a basic science talk than we typically get here, and I think it's going to be pretty good. So my practice typically involves, as many of you know, treating spinal tumors clinically. But it also involves working in a lab where I have a basic science lab. And so in that lab, we study tumor metabolism and intervertebral disc degeneration. And really the sorry, I'm having a little trouble with the keyboard right now. The theme of all of these things in my life, both in the clinic and in the lab, is teamwork and collaboration. And so I put up this picture here of Jason Williams and Chris Weber, just as an example. Many of you know Chris Weber made multiple all-star teams, and Jason Williams did not. But Jason Williams was a fantastic passer, and I think many of us are making all-star teams or doing things, standing under the rim, waiting for the ball, and don't always acknowledge the people that are making things happen. And so many of you that have a lab or have a busy clinical practice or other things know that it takes, somebody like that in each area. And so in my case, Hannah's my admin who really, runs all things in my life, including my office life, Krita in the clinic, and then in the lab, it's Shambhavi. And so Shambhavi started actually as my tech in the lab and was so good and independent and had all these great questions and was a great technician. In addition to being a great scientist, she actually got accepted to our graduate program here at WashU, which is incredibly hard to get into, and is now about done with her PhD. And so what do we do in the lab? Well, the two main areas are disc degeneration and tumor metabolism. And yeah, how did we get to studying tumor metabolism and what are the things we care about? Well, clinically, I'm showing you a picture here of a primary tumor in the pelvis. And anytime you have a primary tumor, as many of you know, you want to do an en bloc resection when possible, meaning you want to get margins all the way around the tumor and you want to get the whole thing out and really cure the patient. That's the goal, that's the idea. But the question arises, what if that's not possible? What if it's around critical structures to where you can't do that and you have to pursue another strategy? Or what if there's already micrometastatic disease that maybe you can't recognize because our tools aren't sensitive enough? But you wouldn't want to do a massive en bloc resection if they had disease that had already escaped that area, right? And so the question is, are there other treatments that you can use to target the tumor in cases like that? So for example, here's a 55-year-old, what I just showed you, with a pelvic sarcoma, underwent a en bloc resection, had good margins, but he had metastatic disease in his chest within a year. And if you look back, it was thought to be there probably the whole time, possibly as micromets early on. Or this patient, a 67-year-old with a chordoma, and it's around both of her vertebral arteries. A little hard to see on the MRI here, but it was circumferential around both verts. And so she did undergo an en bloc-ish type surgery, as we say, with plans for high-dose radiation around those verts afterwards. But the thought remains that we'd like another way to target some of these tumors. And so one of the questions we had early on was, is there anything metabolically unique to tumors that we could target? Of course, if you go back to even the 1920s and 30s, Otto Warburg described what at the time was thought to be a very unique characteristic of tumors. And he said he described what eventually became known as the Warburg effect. And that is that regardless of oxygen tension, tumors like to consume glucose and produce lactate. And so here we see non-tumor tissue traditionally thought that when there's oxygen there, there'll be some lactate production, but a lot of oxidative phosphorylation. And if there's no oxygen, then there's no oxidative phosphorylation, and it's all production of lactate. And he described how in tumors, there seems to be heavy use of glucose, heavy lactate production, despite the fact that there's plenty of oxygen around. And this is something we see in PET scans, for example, where we see that some tumors are very glucose avid and produce lots of lactate. Now, lactate itself has been the subject of controversy over really the last century, if not more. And at times people have said, well, lactate's produced because of hypoxia. People have blamed fatigue on lactate. People have said lactate's a waste product. It's all it can do is go back to the liver and be recycled. And all these things we know are erroneous now, years later. And so why did those things come to be? Why did those things get said? Well, a lot of that comes out of how lactate was really discovered. And so even in the early 1800s, it was noted to be in high concentrations in these worn out, tired, hunted stags by Berzelius. And then later, these experiments in the early 1900s found that lactate was high when there was no oxygen, found that lactate was high, after contractions basically failed and the muscle fatigued. And so that really set the stage for a lot of what was thought about lactate for many years after that. Of course, in the last 20 years or so, in fact, it's been almost 20 years since this article was published. Bruce Gladden and George Brooks have been two of the big pioneers in lactate metabolism. And there's a big story in the Times in them back in 2006 about how this was kind of one of the classic mistakes in the history of science, that lactate's actually, the way that metabolism's coordinated and not a bad thing at all. And so how did that come to be discovered, or how did we find that out? Well, that started with Wendell Stainsby at the University of Florida, who in the late 60s published the first study showing that the contracting lactate releasing muscle wasn't hypoxic at all. It had plenty of oxygen available to it, but it was releasing lactate. And in fact, we found out that hypoxia itself rarely, if ever, is the cause of a high arterial lactate in a whole animal. In fact, if you take humans and you make them do a isolated single leg kick, which is what you're looking at here, and until their muscle fatigues, and this was using near infrared to measure PO2. What you see is that even at the fatigued concentrate, even when the muscle's fatigued, even down to the level of the myoglobin, the PO2 is still over 3. And if you look at humans breathing a hypoxic 12% mixture, it still only gets down to 2. And we know that at the level of the mitochondria and the cytochrome turnover, it takes about 0.5 or less to really limit the passing of electrons. And so even at the most fatiguing hypoxic time, it doesn't limit the actual use of oxygen in the mitochondria. If you look at animals and you cut off the blood supply to part of their intestines, and so this was an inclusion of the SMA in pigs, their arterial lactate actually doesn't really change concentration. And so it's very hard to get an arterial lactate that's elevated from hypoxic event. What's not hard is to see that blood lactate concentrations typically correlate with epinephrine and norepinephrine. So here you see different work rates, and you see the blood lactate corresponding with those. And if you plot the level of epinephrine or norepinephrine, you can see a similar thing that goes along with it. This makes sense because there are receptors on the muscle that norepi can bind to, which then leads to glycogen breaking down into lactate, which can then be released into the blood, which is something we see. And in fact, this is the way that in times of fight or flight, glycogen that's stored in muscle is able to get out into the blood and be used as a fuel. So if you remember, it cannot get out as glucose once it's in the cell, but it can get broken down to lactate. That lactate can then circle throughout. And so we've seen this concept proven over and over now in the last 40 years, where lactate is a great fuel for tissues. We see it in whole animals with carbon tracing studies. We see it with animals run to exhaustion. This is work from Avital Schur, Louisville, where he took hippocampus slices and removed all the glucose and then gave them back lactate, and they recovered appropriately. We've seen it now in heart, we've seen it in brain, we've seen it in spinal cord. A whole host of different studies now demonstrate this use of lactate as a fuel, as a great fuel. We see in newborns that they have a high lactate and high lactate transporters, often thought because of the immature liver at that time that they're relying more on lactate than we do later on. And we see patients that sometimes get critically ill, and actually lactate seems to be somewhat life-sustaining in many of these cases. And so here's a case that we ended up writing up. It was a case of Dr. Nyson's in the Netherlands, where a patient had a glucose of 16, yet they were walking and talking, and in fact their lactate was 25, and later was thought that that was the way they were kind of staying awake and alive, was using that lactate as fuel. You've seen studies like this in bigger journals in the last couple of years, where when you look at lactate and where it goes, you can use tracer studies and find that a lot of it shows up in the TCA cycle being used as a fuel. And so we know now, years later, really out of the muscle literature, that it's the way metabolism in the whole body is coordinated. George Brooks described this with the cell-to-cell lactate shuttle, where lactate-producing tissues release lactate into the blood, where it can then circulate not only to nearby tissues, but tissues throughout the body to be used as a fuel. The only other point I want to make about that is that glycolytic tissues, meaning tissues that are producing lactate, can switch to consuming lactate. Will do so when the blood lactate gets to even above 4 millimolar. And here, you're looking at arterial lactate on the X, and you're looking at lactate balance on the Y. And so 0 means neither consuming nor producing. And what you see is that as you get above 4 millimolar, even the most glycolytic tissues will start then consuming lactate if the concentration's high enough. That's going to come back to play in a little bit here. So we know it's a great fuel. We know it's transported via these MCTs, which you're going to hear more about from Shambhavi. We have since learned that it's also a great signal. And so here's kind of the normal type of metabolism, where if you cut off oxygen and make a cell hypoxic, you're going to get a buildup of pyruvate and lactate, stabilizing HIF1 alpha, leading to VEGF and angiogenesis. But we now know that you can do the same thing in normoxia with importing lactate. So you can import lactate, have the same pathway leading to HIF1 alpha stabilization and angiogenesis. And so how does all this relate to tumor metabolism? Well, one of the early ideas that we had was that, and when I say we, I mean Dr. Nyson in the Netherlands, was that, well, if most cells like lactate and if most tumor cells are producing lactate, perhaps we can make an animal very hypoglycemic and provide lactate for the other tissues of the cell in the event that we can starve the tumor cells. And so we did a variety of things over this. This is years ago now, where we took animals, in this case dogs, and we made them hypoglycemic. You see kind of the setup here on the right. We made them hypoglycemic and then provided them a high level of lactate, so a hyperlactatemic clamp at 8 to 10 millimolars, to see what the difference was and how long they could live off of lactate. And so here you see that concept, this lactate-protected hypoglycemia, and some of those results. And so some of those results are summarized here. On the top, you see red is glucose, and you see that the animals made very hypoglycemic, and then lactate's just kept low, and the animal dies soon after. And in this trial, you see that the animals made hypoglycemic only after raising the lactate up to 8 to 10 millimolar, and the animal lives for many hours after that. Unfortunately, we know now it's not that simple, and we know that there's something that's been referred to as the reverse Warburg, and that is that many tumors seem to consume lactate. And in fact, it's not clear exactly when tumors produce or when they consume. And in fact, Greg Semenza, who recently won a Nobel Prize, was one of the first people to talk about this kind of symbiotic relationship and whether tumor cells were producing or taking up lactate. So the Warburg effect, again, might be something that is true, and it depends on the tissue type or the tumor type. But then there's probably a whole bunch of other spectrum of things that happen with tumors, including the reverse Warburg, where tumor cells are taking up lactate, and then there's probably some in between as well. And so I'll kind of conclude, I think, my part talking about some of the work we did when I was a resident. This is at the University of Utah with Dr. Kevin Jones and Mario Kopecky's lab. Mario Kopecki, of course, was the Nobel Prize winner for the knockout mouse. And we worked on alveolar soft part sarcoma, a unique type of sarcoma with a known translocation. So we took that translocation and we put it into the mouse. And when we did that, you get some leak of expression of that translocation throughout the mouse. And so that's what we did. And what we found is we were able to make tumors that looked exactly like the human tumor. So the mouse grew tumors that looked just like the human alveolar soft part sarcoma. So here you're seeing mouse tumor on the top, human tumor on the bottom. The only difference between this study and work they had done in the lab before I got there was that in the other models where they knocked the gene in like that, they got expression of tumors showing up different parts in the body. And in this particular one, it only showed up in the cranial vault. And when you looked in the cranial vault, it was in different tissues in the cranial vault, but only in that cavity, in that space is where they found them. So we got thinking, why is that different? And what we found is that was the area of the highest lactate concentrations in the animal. And when you look at these tumors, some of the work by Ladani and others showed that they have lots of these MCTs and its chaperone protein CD147 in these granules that you see throughout the cell. And so furthermore, you see that they have a O2 consumption that goes up when you give them lactate. And so it certainly seems like these were cells that were using lactate. So they're probably over here on the reverse Warburg spectrum of things. So here you see how vascular they are, lots of vascularity here, lots of HIF1 alpha. But interestingly enough, when you stain for, so this is a stain actually for hypoxia, they are not very hypoxic at all. And so typically, on the right here, like in the kidney, you'll see areas of hypoxia is the same area where you get expression of HIF1 alpha. And in this case, we didn't have really any hypoxia and lots of HIF1 alpha thought to be, in this case, from the high lactate concentrations. One of the things we did to test exactly that was we took these mice and we gave them injections of lactate compared to just sodium chloride. And what we found is that their, vascularity went up here. It's markers of proliferation, anti-Ki67. Here you see the vascularity on the left, markedly different with the lactate injection. So certainly seem to play a big role. So in this tumor, instead of having hypoxia driving angiogenesis, you now have a situation where it's lactate driving angiogenesis, even in normoxic conditions. And so we started thinking about how could we block this or inhibit this. And we started thinking about, so here's the publication and the things we learned. We started thinking about how to block this or inhibit it. And one of the things we started looking at was these lactate transporters. And so you're going to see more work in a second on these MCTs, but I just show it here to say that it was early on, we had a difficult time doing anything with this, even though we tried different types of things to inhibit MCTs. So here's actually just some failed experiments, basically. So that led us to looking at osteosarcoma for reasons you're going to see in a second here. And, I'll let Shambhavi talk more about it, but, reasons that have to do with patients with these MCT4 expression that's higher doing much worse overall. And so with that, I'm going to stop my talk there, and I'm going to let Shambhavi pick up with kind of some of the work that's been done in osteosarcoma.
Is everyone able to see my screen? My slideshow?
Perfect.
Okay, thank you. Thank you so much, Dr. Goodwin, for the really good introduction of lactate and how it's important for physiology in general, as well as in cancer. Hi, everyone. My name is Shambhavi Bhagwat. As Dr. Goodwin mentioned, I'm a PhD student in his lab. So here goes. So cellular metabolism is a flexible network that allows tissues to meet demands for homeostasis and growth. Now, in cancer, malignant cells acquire metabolic adaptations in response to a variety of cues, both intrinsic as well as extrinsic. Intrinsic factors include characteristics of the parental tissue, as well as new properties arising in the malignant cells that includes their genotype or perturb signaling and gene expression. Whereas extrinsic factors include metabolic stress in the tumor microenvironment, which includes, and which is important for our talk here today, an abnormal milieu of metabolites. And it also includes metabolism of the patient, which is influenced by genetic factors, pre-existing metabolic disorders, or their diet. The metabolic needs of the cancer evolve throughout its progression. Early stages of the tumor growth require nutrient uptake and biosynthesis of certain amino acids or other metabolites. And additional subtype-specific metabolic needs also emerge in locally invasive cancers. Tumors acquire dependence on new pathways during later stages of cancer progression, particularly in metastases and therapy resistance. Most cancer metabolism research is focused on the phenotypes that are observed in clinically detectable tumors or experimental models that arise from them. The terms cancer metabolism and metabolic reprogramming are therefore usually taken to indicate a shared set of pathways that are observed in highly proliferative tumors. Now, as Dr. Goodwin described, the Warburg effect is a prime example of metabolic reprogramming. It was discovered by Otto Warburg. And as we already saw that the cancer cells perform glycolysis or they produce lactate irrespective of availability of oxygen, or I guess unavailability of oxygen. He showed that cancer tissues use copious amounts of glucose to generate lactate, even in the presence of oxygen, and therefore concluded that cancer cells cause an injury to respiration, which is a prerequisite for the transformation of differentiated cells into a proliferative cancer type. Now, everyone here probably already knows that osteosarcoma is a PET-positive tumor, which means that it has a heavy uptake of glucose. Now, glucose is predominantly metabolized via glycolysis, which produces either pyruvate or lactate. Lactate, as again described, was previously known to be a byproduct, but there have been numerous studies showing its importance in different diseases, and in this case, even in osteosarcoma. Here, osteosarcoma patients were seen to have higher lactate in their serum as compared to healthy subjects. And also, patients with higher LDH, which is the enzyme that converts lactate to pyruvate and vice versa, patients with higher LDH had worse survival probability than patients with lower serum LDH. Now, lactate's presence in the cell can either be from glycolysis or it can be from uptake from the microenvironment. Its transport is carried out by MCTs or monocarboxylate transporters. Of the 4 types that are responsible for lactate transport, MCT 1 and 4 are more ubiquitously present in almost all cell types. MCT 1 is considered a major importer of lactate, while MCT 4 is considered a major exporter of lactate. And their expression differs in the so-called glycolytic versus oxidative cells. In osteosarcoma, MCT1 expression has been correlated with the aggressiveness of the tumors. Higher MCT1 expression can be seen in grade 3 tumors as compared to grade 1. And in addition, the survival probability of patients with higher MCT1 is worse than the patients with low MCT1 expression. So One in vitro study back in 2014 demonstrated the metabolic crosstalk between osteosarcoma cells and stromal cells in the tumor microenvironment. They showed that conditioned media from the osteosarcoma cells metabolically reprograms the MSCs or stromal cells. The expression of GLUT1, which is a glucose importer, and lactate exporter MCT4 was upregulated in these activated cells. This suggested that these activated MSCs are highly glycolytic due to the influence of osteosarcoma cells. Further, when they transferred conditioned media from the MSCs onto the osteosarcoma cells, they observed an increased expression of the lactate importer, MCT1, and concluded that osteosarcoma cells likely take up this extracellular lactate when it's readily available. In our lab, we confirmed that osteosarcoma cells indeed at baseline had a much higher expression of MCT1 as compared to HFOB, which is an immortalized osteoblastic cell line. Now, given that the expression of MCT1 is higher in osteosarcoma, we hypothesized that uptake of lactate would be higher in these cells, and inhibiting this uptake by inhibiting the function of MCT1 by either knocking it down with siRNA or with an inhibitor, which is AZD3965, would result in lower or decreased intracellular lactate. And we did actually see that. We saw that intracellular lactate levels were significantly lower when MCT1 function was inhibited. So that concluded that, that study concluded that MCT1 is likely the major importer of lactate in osteosarcoma, and that lactate is important for these cells. Further, to study the overall effect of MCT1 inhibition on tumor progression, we did a pilot study wherein we established an orthotopic model by injecting osteosarcoma cells onto the tibia of athymic nude mice. These mice were then randomly separated into two groups, one control and then the other treated with the AZD3965. These mice were treated orally starting on day 4 post-tumor injection. The mice were weighed and tumor measurements were done every 3 days by caliper. We also did weekly in vivo and micro CT scans. And then the experiment was ended at day 20, at which point the mice were euthanized and then the tumors were excised. Although we had expected to see some bony tumors in the micro CT scans, we were only able to see the tibia. But as you can see, the scans revealed that by the end of the study, the tibia of the mice in the control group were highly disfigured as compared to the mice in the treated group. In addition, we also saw a significant decrease in the tumor volume of the mice that were treated with the MCT inhibitor. Thus, blocking lactate transport via MCTs slows down the osteosarcoma tumor growth in vivo, and dual target therapy would likely prevent the increase in tumor growth that you can see post day 15, by which day the cells had likely adapted to the loss of lactate uptake via other mechanisms. Now that we had established that lactate is taken up by osteosarcoma cells, we were curious about its effects how it affects the cancer cells internally. Given that lactate oxidation occurs at the mitochondria, we wondered whether the mitochondria in osteosarcoma cells are any different than normal cells. And a dive into the literature revealed that osteosarcoma cells they have higher mitochondrial mass, as well as higher activity, which is measured by oxygen consumption rate that directly correlates with ATP production. Mitochondria are highly dynamic organelles that are constantly undergoing fission and fusion. The dynamics are regulated by many proteins. However, OPA1 and mitofusins 1 and 2 are the main regulators of mitochondrial fusion, whereas DRP1 is the main regulator of mitochondrial fission. Interestingly, another paper suggested that when mitochondrial fission in osteosarcoma cells was artificially upregulated, it activated an apoptotic pathway. So that led us to question whether lactate influences mitochondrial dynamics by suppressing fission. The idea that lactate could play a role in mitochondrial dynamics is supported by studies in other cell types. For example, in primary hippocampal cells, the presence of high lactate in the medium induced the expression of mitochondrial fusion proteins, that is MFN1 and 2, whereas the expression of the fission proteins DRP1 and another one FIS1 were downregulated. So to test whether fusion is beneficial to osteosarcoma, we knocked down fusion protein OPA1 and observed that proliferation rate of these cells dropped significantly. On the other hand, when we knocked down the fission protein DRP1, there wasn't a similar significant drop. We also saw differences in metabolic phenotype of these cells after the knockdowns. So absence of DRP1 increased the activity of the mitochondria and increased ATP production. Thus, we inferred that knocking down the DRP1 would lead to hyperfused mitochondria, which are more beneficial to osteosarcoma cells. To confirm whether our knockdowns actually made the changes to the mitochondrial network as we had expected, we used confocal imaging to look at the mitochondria in the cells. Here, the orange stain or represents the mitochondria, whereas the blue stain represents the nuclei of the cells. And as you can see, when the fusion protein OPA1 was knocked down, the mitochondria were not able to fuse and they were more short and fragmented. Whereas when the DRP1 was knocked down, which is a fission protein, mitochondria were more elongated and looked like they were more fused. Further, we wanted to see whether the effect of MCT1 inhibition, so inhibiting the uptake of lactate, would affect mitochondrial network. And we found that in the presence of the MCT1 inhibitor AZD3965, mitochondria in these cells were very fragmented and rounded, which helped us conclude that lactate does somehow affect mitochondrial dynamics and that blocking its uptake creates an imbalance in fission and fusion. So to summarize our findings so far, we can say that lactate uptake does occur in osteosarcoma cells and that they have higher expression of MCT1, which is the lactate importer. Mitochondria in osteosarcoma cells are different than in normal cells, and that lactate plays a role in mitochondrial dynamics and thereby helps in osteosarcoma progression. And that brings me to our theory that extracellular lactate is taken up by osteosarcoma cells via MCT1, resulting in an imbalance of mitochondrial dynamics, likely by upregulating mitochondrial fusion and thereby promoting osteosarcoma proliferation. And inhibiting this uptake would be a good targeted therapy for osteosarcoma. And with that, I would like to thank everyone for attending this talk, and to all the members, former and current, of the Goodwin Lab, and everyone at WashU who's played a role in shaping this project. And thank you. We're here for any questions.
Well, Shami, that was fantastic, like usual. I don't see any questions in the chat, so I'm going to ask you a question, even though I ask you questions every day. So you have spent a lot of time with a lot of different types of tumor. And I don't think I said this at the start, but when Shami started, she actually worked with multiple different tumor types, multiple different cell lines, primary and metastatic, in a lot of our early work. And so I guess my question is, how much of what you just showed us with osteosarcoma metabolism is going to be or is similar to other cell lines, or do you think osteosarcoma is a big, unique outlier in its behavior?
Um, from what I have worked on, it every cell type is different. And like I said, like for example, like in osteosarcoma, we know that MCT1 expression is pretty high. And that does not necessarily mean that other cancer types have, similar cellular phenotypes. So yes, there are certain cancer types which do behave in a similar way, wherein they take up lactate, whereas some other tumor types are more glycolytic. Is that
I knew you were gonna say that each was different. Um, the which we see even in our different osteosarcoma cell lines, right? So yeah, it's I understood that.
yes?
How much do you think the lactate concentration in these different protocols matters? And and uh, and maybe you can comment on what you think about lactate concentration in human tumors is.
In sorry, what was the last part again?
Yeah, in human tumors, like the tumor microenvironment.
Well, a lot of the literature does, and we have talked about it in the past, that a lot of literature uses really high lactate concentrations for their in vitro studies, which I don't think is physiologically possible to sustain. So if you're talking about like a specific number, is that what you were asking, Dr.
No,
Goodwin?
no, just exactly what you're saying, kind of in general. I mean, it seems like you can, that's exactly what I'm asking is kind of whether all the studies we see are applicable or not, or, sometimes
Really
it's kind of
hard to it's really hard to deduce science, I guess, based on just in vitro studies, because in vivo it's completely different. Like you cannot, I don't think you would be able to see any tissue type that has 50 millimolar of lactate in the microenvironment. And so I guess that's why it's really important to have in vivo or, clinical trials for this matter. It is, again, from my experience, it is a little hard to actually test the concentration of lactate in the tumor microenvironment because there are certain probes that you can just use and like inject into a tumor. But then at that point, you don't know if you're actually injecting it in a cell or whether it's just the tumor microenvironment. So that's still like a topic of debate, in my opinion. But again, in vitro studies with really high concentration of lactate, I don't think they're reliable or they're even comparable to what actually happens in the body.
All right, I'll stop torturing you in front of everybody. My last question is about potential translation of the work and next steps and that sort of thing. You think the future it's more of an opinion question, probably more in line with things like clinical trials for MCT inhibitors? Is it more in line with doing something with the fusion fission proteins? Is it changing concentrations? Is it using something in concert with other chemotherapeutic or immunotherapy drugs? What are your thoughts on some of that?
So MCT1 inhibition is definitely a very popular target, and the drug that we just talked about, AZD3965, has been in clinical trials for Burkitt's lymphoma, I think, and I think they reached like level 2, 3, and it failed after the third one, and it has failed in the past for solid tumors. And so that makes me think that just using a singular target is not enough. It's enough to slow down the tumors, but not enough to completely be used as a treatment strategy. It has to be combined with something else. And it's really hard to find targeted therapy for mitochondria because, well, they're present in every single cell type. So I guess there has to be another way or mechanism wherein you're targeting only the mitochondria in these cells, like, for example, like nanoparticles that are directed to the cancer cells, or like CAR therapy, which somehow targets the mitochondria of cancer cells.
There's another question here in the box that says, great presentation. Spinal cord itself presents a unique metabolic environment with different, for example, pH, nutrient availability, mechanical pressure compared to the brain. how much of the metabolic reprogramming we see in spinal tumors is an intrinsic property of the tumor cells versus an adaptive response to these environmental constraints. So I think it's an interesting question because I don't. I'll let Shamavi answer it or talk about it, but the I think here you're saying spinal cord tumors, which most of I got to think of what all we looked at in the early days. But most of what we looked at has been not tumors intrinsic to the cord itself. Um, but that being said, there are plenty of kind of constraints even in the areas around there. So, Shambhavi, I don't know if you have any thoughts about this in terms of intrinsic properties of some of these tumors versus adaptive responses. I think you probably do have thoughts on that.
Um, with spinal tumors, I would have slighted this question to you because you're obviously the expert. Um, no, but like what you said,
Well, I think I mean, I guess my answer would be that I think it's probably, I think it's probably some of both. I think one of the difficult things, particularly with um, using a metabolic approach, is how adaptive these cells can be. And so that's actually part of that. The last question and part of one of the things we talk about is, how much a lot of these cells appear to be able to adapt to their constraints, even in extreme constraints. And so they seem to be very adaptive and very resourceful. And so I think it's some of both. I think teasing out how much of each is obviously pretty difficult. Well, Shamavi, thank you again. Excellent presentation, and I want to thank everyone that's here tonight, and for the great questions as well. And I think with that, we will conclude. Thanks.
thank you.